Hi everyone. This is the second lecture for your chapter Respiration in Plants. In the last class, we studied a little bit about the living system and how cellular respiration helps you or helps a cell survive. Okay, and what is cellular respiration? How is it comparable to combustion? What kind of process it is? And is oxygen always required for the process? So we ended the lecture saying that no, oxygen is not always required for respiration. There is a type of respiration called anaerobic respiration, which does not require oxygen at all. So we can oxidize glucose in absence of oxygen. And this pathway is called glycolysis. So today's topic will be glycolysis. Glycose means sugar, and lysis, anywhere where you read lysis, it means splitting. Okay, you're breaking something down. What are you breaking? You're breaking glucose. So glycolysis. So this glycolysis, the process, it was deduced. Deduced meaning somebody who discovered it. Okay, somebody who found out the pathway. So who did this process? It was done by Gustav Embden, Otto Meyerhof, and J. Parnas. So there were three people who found out the whole process. Okay, Embden, Meyerhof, and Parnas. Using their first names, okay, or initials, the glycolysis pathway is also known as EMP pathway. Embden, Meyerhof, Parnas. So if somewhere if instead of glycolysis it is mentioned EMP, do not be worried. It is the same pathway. Okay, fine. So it occurs in the cytoplasm. Okay, after knowing what the process is in general, it is just breaking of glucose in absence of oxygen. We also saw who discovered it. The third thing which is important is where does this process take place? Okay, where is this anaerobic respiration taking place? So it takes place in the cytoplasm of all cells and in all living organisms. Clear? Okay. In this process, the glucose undergoes partial oxidation. Remember this term. If the glucose undergoes complete oxidation, you get carbon dioxide as the product. However, if the glucose undergoes partial oxidation, you get two molecules of pyruvic acid as product, right? So this is the whole process. You have a molecule of glucose, it will undergo glycolysis, and it will give you two pyruvic acid or pyruvate molecules as product. In plants, this glucose and fructose are derived from sucrose. Okay, we shall be studying transport very soon. So, just to give you a quick revision, you must remember that sucrose, which is a non-reducing sugar. Okay, we just have completed biomolecules, so you must remember those things. So which is a non-reducing sugar, which means it is least reactive, right? So sucrose is used to transport sugars in the plants, okay, through phloem. So this sucrose, wherever it is required, okay, wherever it needs to be respired, it will be divided into glucose and fructose using an enzyme which is known as invertase. Clear? So this mixture of glucose and fructose, if you know, it is also known as invert sugar, right? So invertase is used to break down sucrose into glucose and fructose. Now these two monosaccharides, now you must also know this, right? Then glucose and fructose are nothing but they are isomers of each other. They are functional group isomers. So you can convert all kinds of monosaccharides, okay, hexoses, into glucose. So there can be galactose, which can be converted into glucose or it can enter glycolysis, fructose, mannose, okay, all of these hexoses can enter the glycolysis pathway. After entering, now we will just consider glucose, but as I already told you, any of these hexoses can enter the glycolysis pathway. Let us study it, okay? Basically, the whole process, it is not as simple as that glucose gives you 2 pyruvate. No, actually this whole pathway is divided into 10 different steps. Okay, it happens in 10 steps. I hope you remember that respiration is the same process as combustion. Okay, it is also oxidation, but it occurs in slow stepwise manner, so that at every step you are conserving energy, okay, or you are converting it into ATP. Chalo, let us see. First, we will be seeing step number 1 to step number 5. These first 5 steps of glycolysis are known as the stage where you actually use ATP. Okay, you are not producing ATP, but you are using ATP. Clear? So the first step is, here is your glucose. Okay, glucose undergoes phosphorylation. So there is a phosphorylation reaction. What does phosphorylation mean? It means addition of phosphate group. Clear? So glucose is phosphorylated by an enzyme called hexokinase. Wherever you hear the word kinase, remember this much. Okay, it is an enzyme which is related to ATP. Okay, it will be related to ATP getting converted to ADP. Clear? And second thing, wherever there is ATP and kinase involved, there is always magnesium ion which is present. Clear? Magnesium ion does what? It actually balances out the negative charges on the phosphate. Okay, so it is helping in that process. Okay, so this is the first step of glycolysis. Glucose, by action of hexokinase enzyme and use of one ATP, it gets phosphorylated. Where is the phosphate getting attached? It is getting attached on the 6th position. So from glucose you get glucose 6 phosphate. This reaction is not reversible. Okay, it is irreversible. Why? Because you are using energy for this process. So it is endergonic reaction. You are using energy. Clear? So wherever, whatever reaction where you are in, there is input of energy, it is mostly irreversible. Clear? So glucose to glucose 6 phosphate. Now one more thing. Once you convert glucose to glucose 6 phosphate, this glucose 6 phosphate cannot leave the cell. Okay, it is um, always destined now to stay inside the cell. It cannot leave the cell because of this phosphate charge. Fine. This is the first reaction, glucose to glucose 6 phosphate. The second step is glucose 6 phosphate getting isomerized to fructose 6 phosphate. Clear? So this is the glucose ring. Okay, and 6 phosphate. There is an isomerase type of enzyme. Clear? Remember the 6 types of enzymes. So one of them is isomerase. What a kind of isomerase is this? So it is converting the phosphoglucose. Okay, because this glucose has phosphate group attached to it. So it is phosphoglucoisomerase. Clear? It is isomerizing the glucose phosphate. What can be the other isomer of glucose? So you have functional group isomer called fructose. So glucose 6 phosphate is getting converted into fructose 6 phosphate. So now you have this fructose molecule with the phosphate group attached to it. The third step is this fructose 6 phosphate. Okay, it is, see, just an example why we are doing all this process. Okay, why do we need to add ATP? This is also some questions that I get from the students, like, madam, why do we need to add ATP? We should be getting ATP, right? So if you want to break something, you have to put some energy into it, right? Let us say you want to burn some wood. In case of that, you have to first light a matchstick, right? And then only you should create some spark, then only it will light up. Okay, so same thing, before you can break the molecule, you have to make it unstable. Glucose is a very stable molecule, it will not break easily. But now you want to break the carbon-carbon bonds, you want ATP out of it. So what do? You make it unstable. How do you make it unstable? You add phosphate to it. Clear? So now from glucose you made an unstable sugar which is fructose 6 phosphate. When you add one more phosphate to the process, you get fructose 1,6 bisphosphate. Clear? So now you are phosphorylating fructose 6 phosphate and converting it into fructose 1,6 bisphosphate. Bis because the phosphates are present on or attached on two different carbons. Clear? The enzyme used in this process is called phosphofructokinase. See, kinase comes again, okay, and phosphofructo because this is fructose phosphate that is being used, okay. So phosphofructokinase, ATP is used, and as always, magnesium is also required for this process. Clear? This step is also irreversible. Clear? Because it is using ATP, it cannot go back. So this step is also irreversible. Also, it is the pacemaker. Okay, it will decide the pace of glycolysis. This step will decide at what rate glycolysis should go on. Okay, if you are, let us say, at sleep, you do not require much ATP, then glycolysis rate will be slow. If you are running, okay, then you require more amount of energy, so glycolysis should be more, and hence the rate of glycolysis will also be high. So this pace, okay, this speed of glycolysis, at what rate should glycolysis take place, is decided at this step. Okay, it is decided by the phosphofructokinase enzyme. Clear this much? Okay. So till now we have seen glucose to glucose 6-phosphate, glucose 6-phosphate to fructose 6-phosphate, fructose 6-phosphate to fructose 1,6-bisphosphate. The fourth step is actually when the fructose 1,6-bisphosphate is broken. Okay, you've made it unstable. Now you can easily break the bond. Okay, so fructose 1,6-bisphosphate is broken down into phosphoglyceraldehyde, okay, or 3-glyceraldehyde 3-phosphate. This is the structure. You must know the structure of glyceraldehyde by now. We have just studied it in biomolecules. So on the third carbon there is phosphate attached to it, so glyceraldehyde 3-phosphate. And the other molecule is called dihydroxyacetone phosphate. Okay, you must also know the structure of dihydroxyacetone, right? We saw it in triose sugar. So just add phosphate to it and you get this DHAP molecule. So from fructose 1,6-bisphosphate, now you have two triose sugars, DHAP and glyceraldehyde 3-phosphate. Clear? The enzyme which is responsible for this breaking is called aldolase. Okay, and aldolase, as you can see, it is not adding any water molecule, so it is a lyase kind of enzyme. Clear? 6 classes of enzymes. We have also seen lyase type of enzyme, so aldolase belongs to that lyase class. Now the next step, okay, the fifth step is where there is isomerization again. So for the further reactions you require just glyceraldehyde 3-phosphate. So what is done? Dihydroxyacetone phosphate, which is a ketotriose, it gets isomerized into aldotriose, which is glyceraldehyde 3-phosphate. Here, so DHAP gets converted into glyceraldehyde 3-phosphate. So this is your fifth step. Okay, and the enzyme here is isomerase. Clear? Because it is converting the ketotriose into aldotriose. That is the only functional group isomerism that is involved here. So these are your first 5 steps, and during this first 5 steps are 2 steps. Glucose to glucose 6-phosphate you used 1 ATP, and fructose 6-phosphate to fructose 1,6-bisphosphate you used 1 ATP. The next steps, okay, next 5 steps are known as payoff phase. Payoff meaning you are getting the energy, okay, out of these reactions. You invested with 2 ATPs, now you will be getting back the ATPs. Clear? So we have glyceraldehyde 3-phosphate molecules. These glyceraldehyde 3-phosphate molecules are phosphorylated by inorganic phosphate. Here it is important to remember there is no ATP involved, okay? It is inorganic phosphate, so H3PO4 2 minus. Okay, from that there will be PO4 2 minus, which is being added. Clear? So this is your glyceraldehyde 3-phosphate. This is your phosphate, inorganic phosphate PI, which is added. The enzyme is known as glyceraldehyde 3-phosphate dehydrogenase. Now from the name you know dehydrogenase means to remove hydrogen. Okay, so this hydrogen is removed from glyceraldehyde. So what does that mean? That means the glyceraldehyde is oxidized. Clear? And what is it oxidized into? I had told you, if you oxidize aldehyde, you get carboxylic acid. Clear? Always remember these reactions. I know you must have even understood them in chemistry. But if you have alcohol, so OH, clear? Then you have aldehyde, which is CHO. Or you can even have ketone, which is C double bond O. Okay. And here there will be R, of course. And then you have carboxylic acid, which is this structure. Okay. Fine. If you reduce carboxylic acid, you get either ketone or aldehyde. Mostly you get aldehyde, clear? And if you reduce aldehyde, you get hydroxylic group, okay, OH group. Same way, if you oxidize OH, you get aldehyde. If you oxidize aldehyde, you get my mistake, carboxylic acid, okay? Aldehyde to carboxylic acid is oxidation. Carboxylic acid to aldehyde is reduction. This process of reduction we saw during photosynthesis. Here we are studying oxidation of glucose. So there will obviously be oxidation. Okay. So aldehyde, 3-phosphoglyceraldehyde gets converted into 1,3-bis-phosphoglycerate. Clear? Let me clear that. Okay. So glyceraldehyde 3-phosphate dehydrogenase is the enzyme. You are adding 2 more phosphate groups. Okay. Apart from that, you are oxidizing this molecule. So from glyceraldehyde, it is getting converted into glycerate. Okay. So 1,3. So now on the first carbon you have phosphate. On the third carbon also you have phosphate. So 1,3-bis-phosphoglycerate. Clear? From glyceraldehyde 3-phosphate, 1,3-bis-phosphoglycerate. During this process, the hydrogen that are removed from glyceraldehyde are added to NAD+. So you get NADH plus 2H plus. Clear? So here you are getting reducing power. Okay, NADH. Always remember, in case of photosynthesis, the reducing power is NADPH, and in case of respiration, the reducing power is NADH. Clear? Okay. I hope till here everything is clear to you. Glyceraldehyde 3-phosphate to 1,3-bis-phosphoglycerate. Now this 1,3-bis-phosphoglycerate is converted into 3-phosphoglycerate. Okay, you have just added one phosphate. Now you are removing that one phosphate. Clear? So this process happens with help of an enzyme called phosphoglycerate kinase. As I already told you, kinase means there will be ATP which is involved, and magnesium ion will also be present. From this 1,3-bis-phosphoglycerate, you are removing the one phosphate group and adding it to ADP, okay, adenosine diphosphate. Now this adenosine diphosphate will become ATP, adenosine triphosphate. So you are generating one ATP at this step, okay, at step number 7. Clear? And the product is just 3-phosphoglyceric acid or 3-phosphoglycerate. This process of generating ATP, okay, during such kind of reaction is called substrate level phosphorylation. Okay, substrate level phosphorylation. We have seen photophosphorylation. This is a new type of phosphorylation. It is called substrate level phosphorylation. The 8th step is this 3-phosphoglyceric acid or 3-phosphoglycerate. It gets converted into its isomer, which is 2-phosphoglycerate. What is the difference between the two? The only position of phosphate group is changing from third carbon to second carbon, right? So 3-phosphoglyceric acid is isomerized to 2-phosphoglyceric acid or 2-phosphoglycerate, and the enzyme used here is phosphoglycerate mutase. Mutase is just a type of isomerase, fine? The next step is the 9th step. Here the 2-phosphoglycerate, which is this molecule, okay? So here you have 2-phosphoglycerate. It will get dehydrated. There is dehydration. Okay. What does dehydration mean? You are removing one water molecule. So this OH plus this H plus will be removed in form of water. If these OH and H they get removed, then these two carbons have an extra valency. What will they do? They will form a double bond. Clear? So from 2-phosphoglycerate, you are getting phosphoenol. Enol, why? Because there is a double bond. So phosphoenol pyruvate. Clear? Phosphoenol pyruvate, okay, or pyruvic acid. This is the 9th step, and the water molecule is removed here. The enzyme is enolase. And the final step is another step where you are getting ATP, is phosphoenol pyruvate by help of pyruvate kinase. Okay, the enzyme is pyruvate kinase. It will add a phosphate to this ADP and get converted into pyruvate only. this phosphate is removed from the group. Clear? So this here you get the final product, which is pyruvate and one molecule of ATP per molecule of PEP. Phosphoenol pyruvate also we have seen in C4 pathway, right? This step is again irreversible. All the steps where ATP is included are irreversible. So this is the whole process, okay, the whole reaction of glycolysis. Glucose to glucose 6-phosphate, glucose 6-phosphate to fructose 6-phosphate, fructose 6-phosphate to fructose 1,6-bisphosphate. So you added two phosphates here, okay? You used 2 ATP during these reactions. Fructose 1,6-bisphosphate will be broken down in the middle and give you DHAP and glyceraldehyde 3-phosphate. DHAP is converted into glyceraldehyde 3-phosphate. So now you have two molecules of glyceraldehyde 3-phosphate. Each molecule will undergo the same reaction. It is just 2x, okay? So 2 times the same reaction. Glyceraldehyde 3-phosphate will become 1,3-bisphosphoglycerate. There is oxidation, clear? So here the from aldehyde you are getting carboxylic acid group, clear? 1,3-bisphosphoglyceric acid will become 3-phosphoglyceric acid. It will give its phosphate to ADP and generate 1 ATP. 3-phosphoglycerate will become 2-phosphoglycerate. This 2-phosphoglycerate will become phosphoenol pyruvate, and this phosphoenol pyruvate will release this phosphate and become pyruvate. This phosphate will be given to ADP and one more molecule of ATP is generated. Remember, this pathway from glyceraldehyde 3-phosphate will all be happening in twice the number, okay, in 2x. So you are generating 4 ATP in total. Here, the phosphofructokinase and hexokinase are allosteric enzymes. Allosteric enzymes meaning they have a separate site where another molecules can come and bind and change their catalyzing power. Okay, you remember we have studied this in case of enzymes in non-competitive inhibition. Okay, so the reaction catalyzed by these enzymes are control points of glycolysis. After hexokinase reaction, you have glucose 6-phosphate, which cannot leave the cell, and after fructokinase, phosphofructokinase acts on this, whatever the fate, okay, of this molecule, which is fructose 1,6-bisphosphate, is to go for the glycolysis only. It cannot go anywhere else. Clear? The first, third, and the last reaction of glycolysis are irreversible. Further, oxidation of pyruvic acid and NADH in mitochondria requires oxygen. So the fate of pyruvic acid is decided by presence or absence of oxygen. Okay. Now what will happen to pyruvic acid? We saw partial oxidation of glucose. What happens after that is decided by presence or absence of oxygen. Right. So just some tally about the ATP. You use 1 ATP each at the step 1, which is hexokinase step, glucose to glucose 6-phosphate, and you are using one more ATP at the third step, which is fructose 1-phosphate to fructose 6-phosphate to fructose 1,6-bisphosphate. Yeah. So total 2 ATP were invested. Then 1 ATP is produced at each step. Okay, step number 7 and step number 10. Step number 7 is 1,3-bisphosphoglycerate getting converted into 3-phosphoglycerate. So you are getting 1 ATP. And at 10th step, from phosphoenol pyruvate, you are getting pyruvate. So at this step also you are getting one more ATP. Total, there are 4 ATP which are gained. Okay, because everything after this step is happening in 2x. Okay, you can see the number 2 ahead of all of these molecules. Thus, the net ATP produced by glycolysis is 2, because 4 you got. Okay, you had invested 2 rupees, you are getting 4 rupees. So your net profit is how much? Your net profit is 2 rupees. Same way here, you invested 2 ATP, you got 4 ATP. So net ATP produced is 2. However, according to your textbook, they are including all of the ATPs. Okay, so just consider 4, that you are getting 4 ATP. Right. Now at the step number 6, you are using 2 NADH, okay, and you are using 2 NAD plus and getting 2 NADH. Clear? This NADH will further be used for oxidative phosphorylation. We will see that at that time. For now, remember you are getting 4 ATP and 2 NADH. There is no use of oxygen. Also, there is no carbon dioxide which is released at this step. Clear? So what happens after pyruvic acid? That was our original question, right? So if there is presence of oxygen, there will be aerobic respiration, and using this aerobic respiration, it will get converted into acetyl-CoA. From acetyl-CoA, there will be Krebs cycle, electron transport chain, and finally you are releasing it in form of carbon dioxide. So there is complete oxidation in presence of oxygen. What happens if there is no oxygen? So in case of absence of oxygen, there will be fermentation which takes place. So without oxygen, there is fermentation. Fermentation can be of two types. There can be alcoholic fermentation where you produce alcohol, or there can be lactic acid fermentation where you produce lactic acid. These pathways, okay, this fermentation and the aerobic cellular respiration, we shall be studying in the next class. So before that, I hope you just refer to the whole pathway, okay. Just understand all the reactions that are occurring, okay, all the ATP we are using, all the ATP we are getting, and the NADH. Never forget the NADH that you get from glycolysis, okay. It is important in this fermentation process. We will understand it at that time. So see you next time.